For this final topic video in Unit 1, topics 1.6 and 1.7 will be addressed: nucleic acids and proteins. Nucleic acids and proteins are two of the four major biological molecules, and although they're structurally and functionally quite different, they're actually probably the most closely related of them all. Found in the nucleus of eukaryotic cells with a below 7 pH making them acidic, nucleic acids carry important genetic information. This information amounts to what is essentially the instructions for constructing proteins. At this point in the year, we're just going to be taking an introductory look at nucleic acids and proteins. But later in the year, we will see them both again in much more detail. As mentioned, nucleic acids are responsible for storing and transmitting genetic information. The primary storage nucleic acid is DNA. A second nucleic acid, RNA, will be reviewed later. In eukaryotic cells, DNA is found stored within the nucleus, and in prokaryotic cells, it's found free-floating in the cytosol in a region called the nucleoid. In both cases, the cell's genetic material is copied so that the resulting offspring cells have a complete, accurate set of genetic information. Both eukaryotic and prokaryotic DNA are typically arranged into structures called chromosomes. However, eukaryotes tend to have multiple chromosomes that are linear, whereas prokaryotic DNA is circular, meaning that the ends of the molecule are connected to each other, and there's often just a single chromosome in prokaryotic cells. The individual monomers of nucleic acids are called nucleotides, and just as with other macromolecules, they are joined together by dehydration synthesis. Now, let's take a look at the structure of a single nucleotide. Individual nucleotides are constructed of three smaller components: a phosphate functional group, a pentose sugar, and one of 5 nitrogen-based molecules. In a much later unit, we will explore the significance of each of those nitrogenous bases. The resulting nucleotide is structured with the 5-carbon sugar at the center, the phosphate group bonded to the carbon in that sugar that's outside of the ring, and the unique base portion bonded to a carbon in the ring. The 5 carbons in the sugar can be identified with a numbering system. The number 5 carbon, more accurately called the 5' carbon, is where the phosphate group is attached. The 1' carbon is where the nitrogenous base is attached. And the 3' carbon possesses a hydroxyl group, which, as we've seen before, is important in dehydration synthesis. This gives a nucleotide an orientation or directionality. In a dehydration synthesis reaction between nucleotides, the hydroxyl group on the 3' carbon and a hydrogen atom from the 5' carbon of another nucleotide are used to form water. In the resulting nucleic acid polymer, nucleotides are joined together by a phosphodiester bond, which exists between the 3' carbon of one nucleotide and the 5' carbon of another. It is important to notice that this polymer also has the 5', 3' directionality, regardless of the length of the polymer. In DNA, which is a nucleic acid comprised of 2 strands of nucleotides, those 2 strands are held together by the presence of hydrogen bonds between the bases. The adenine-thymine base pair forms 2 hydrogen bonds, whereas the guanine-cytosine base pair forms 3. The relationship between DNA and RNA, where DNA provides information for the construction of RNA, which in turn provides information for the construction of a protein, will be explored in much greater detail in a later unit. While it's true that DNA and RNA have more in common than not, there are 3 important structural differences that make it easy to distinguish between the two. First is the fact that DNA is typically comprised of 2 strands of nucleotides twisted around each other, whereas RNA is usually a single strand. The second difference resides with the sugar present in the nucleotides. DNA utilizes the pentose sugar deoxyribose, and RNA has the pentose called ribose. The slight single oxygen atom difference between the two can be observed in these illustrations. The third and final difference is found in the nitrogenous bases that the molecules possess. While three of the bases, adenine, guanine, and cytosine, are identical, only DNA has thymine, and only RNA has uracil. Although they are structurally similar to one another, thymine has a methyl group, making it more chemically stable than uracil. The result is that DNA is better for the long-term stability and storage of genetic information than RNA is. And before moving on, it is worth mentioning that single-stranded DNA as well as double-stranded RNA do exist. These exceptions to the rule, however, aren't known to exist in any cellular organisms, instead only being found in certain types of viruses. Inside a typical cell, the most numerous molecules are proteins. Proteins play a role in practically every aspect of a cell's functions. The monomers of proteins are called amino acids, which are linked together in long chains to form a polymer, a polypeptide. A polypeptide, folded and shaped into a functional molecule, is called a protein. The statement that says, quote, "structure determines function" is ubiquitous in biology and is brilliantly illustrated by proteins. The 20 commonly found amino acids in organisms can be grouped based on their properties. Some are hydrophobic, some are hydrophilic, some are basic, and a couple are acidic. You may recognize in this illustration that all 20 amino acids have a shared structure and a unique structure. The shared components, which are all identical and shaded in sort of a grayish color, is called the backbone and contains the amino group on one end of the molecule and a carboxyl group on the other. The unique portion connected to the central carbon atom, shaded in yellow, green, pink, or blue, is called the side chain, or R group. It's the R group that grants each amino acid its unique chemical properties. This table shows the amino acids in their charged forms called Zwitter ions. However, amino acids also exist in an alternate, uncharged form. Because the amino group can accept a hydrogen ion or proton, it's considered basic. The carboxyl group can give up a proton, making it acidic. Just as with carbohydrates, amino acids are linked together via dehydration synthesis. The resulting bond, called the peptide bond, is what links amino acids together. In order for a protein to function properly, it must assume a specific shape, more technically known as its conformation. The types and positions of amino acids in the polypeptide chain are what determine how the polypeptide will conform into a three-dimensional shape. All proteins have three levels of structure. The first and simplest level is the polypeptide, which is just a chain of amino acids. The shortest polypeptide is generally accepted to be at least 20 amino acids long, and the longest known is a human muscle polypeptide called titin, which has over 34,000 amino acids. At one end is the functional amino group, and at the other is the functional carboxyl group. All polypeptides, regardless of length, have this same directionality. This is called primary structure. Secondary structure involves the formation of hydrogen bonds between different regions of the backbone of the polypeptide chain. These hydrogen bonds can cause some regions of the polypeptide to form a corkscrew-like shape called an alpha helix, while other regions form zigzag structures called beta-pleated sheets. Depending on the length of the polypeptide, there could be numerous alpha helices and beta-pleated sheets that form. Tertiary structure is dependent upon interactions between the R groups of different amino acids. R groups with a positive charge can be attracted to R groups with a negative charge, forming an ionic bond. Due to London dispersion forces, R groups that are hydrophobic will fold in on each other to avoid being in contact with water. R groups can also interact with one another to establish a hydrogen bond. Those R groups that contain sulfur, they can covalently bond to one another, resulting in the creation of a disulfide bridge. Primary, secondary, and tertiary structure are present in every single protein. Some proteins, however, are the result of more than one polypeptide chain associating together. These proteins are said to have quaternary structure. Transthyretin, which is a hormone transport protein, is the result of 4 identical polypeptides joined together. The protein collagen, which is important in many connective tissues like those in our skin, tendons, and ligaments, is the result of 3 polypeptides twisted around each other. Necessary for the transport of oxygen, the red blood cell protein hemoglobin is comprised of 4 polypeptides. So to summarize, primary structure is a chain of amino acids. Secondary structure involves interaction among the backbone. Tertiary structure involves interaction between R groups, and quaternary structure, if present, is the result of multiple polypeptides associating together. Much later on in the school year, we will tackle the process whereby a cell's DNA is used to ultimately create proteins in a process called gene expression. And with that, these 2 topics have come to a close. Take care.